Hello, everyone. Thanks for the introduction. So I would like to thank everyone for tuning in for today's webinar. So today, I will be presenting on the solutions for new variant of concerns and also evaluation of neutralizing antibody for COVID-19 vaccination monitoring. So for today's webinar, I will be covering several topics. So first, I would like to give an introduction on the Omicron variant, and also the protective immunity and neutralizing antibodies. Next, I will be discussing the technology called surrogate virus neutralization test, which is the SVNT, and which is the technology behind the cPass. And for discussing this, I will be covering topics including neutralizing antibody as the correlate of protection. And I will also be talking about the tools that GenScript provides to accelerate your Omicron research. And last but not least, I will also discuss how we can use the SVNT technology to detect neutralizing antibody against variant of concerns. So the last topic I will cover is the clinical significance of evaluating the neutralizing antibody response. So I will be covering how the SVNT technology can be used for next generation vaccine development, and also the neutralizing antibody dynamics and antibody longevity post-infection and vaccination. And lastly, I will also talk about how we can use the neutralizing antibody response to guide the booster dose regimen. So first, I would like to give some introduction on the Omicron variant, which I believe most of you have already heard. So the Omicron was first detected in November last year in South Africa. And until last month, it has already spread to more than 110 countries. So this is the new reported case that is tabulated for all time. So we can see that due to this fast spread of the Omicron variant, it has propelled the global caseload to new highs. So currently the daily average is almost reaching the level of 3 million. So due to this very fast increased transmissibility of the Omicron, so it is displacing Delta in several countries. So when we see the overview of all the variants in South Africa, we noticed that when just before November, the Delta is still the main circulating variant of concern. And since the onset of Omicron, it has already displaced Delta and become the main circulating variant of concern. So this is also the trend for USA right now. So for South Korea, the main circulating VOC is Delta right now. So people have been wondering, so what has caused the Omicron to have such increased transmissibility? So one of the reasons may be due to the increased site of mutations on Omicron. It was reported that Omicron, there are more than 30 changes to the spike protein, and among them, 15 of them is in the receptor binding domain region, which is the main region that interacts with the human cells. And also there are 2 mutations on the M protein, which is the nucleocapsid protein. So next, I would like to share the key research priorities that was identified by the WHO, so that we can come up with solutions to tackle this new variant. So the first research priority is to evaluate the ability of post-vaccination and convalescent sera to neutralize Omicron. So this can be done by neutralization tests. And the second priority is to evaluate the ability of current vaccines to prevent mild or severe disease. So this can be shown by the real-world vaccination protection. And lastly is more focused on the evaluating whether the current treatments towards COVID-19 is still effective. So for today's webinar, I will be focusing more on the in vitro neutralization test that is available for us to measure the neutralizing antibodies. And I will also share how neutralizing antibody can be correlated with the real-world protection data. So just a quick refresher before we go into the neutralization tests. So for the SARS-CoV-2, when the virus come into contact with the cells, the spike protein on the surface of the virus will bind to the ACE2 receptor that is expressed on the surface of the cells. So once it binds to the ACE2 receptor, it will trigger the internalization of the virus into the host cell, causing the infection and also illness. So when we zoom in into the area, we find that this region, which is highlighted in green here, which is the spike RBD region, the receptor binding domain of the spike protein, abbreviated as the RBD region, is the region that specifically binds to the ACE2 receptor. So when our body is infected or when receive vaccination, we will develop adaptive immunity that can protect us from future infection. So for the adaptive immunity, it consists of two major arms. The first arm is humoral immunity, which is mainly mediated by antibodies. And the second arm is the cell-mediated immunity. For the cell-mediated immunity, T cell is the mainly play the main role in this. So the T cells are activated after the virus enters the host cell, and it can kill the infected cells to stop the viral replication. And hence, T cells is key in protection against severe disease. And we also have the humoral immunity, which is mainly mediated by antibodies. So antibodies are the ones that act before the infection takes place. So according to the different function of the antibody, it can be categorized into binding antibody. So the binding antibody binds to the virus, but may not interfere with its infectivity. And we also have the neutralizing antibodies, which by definition, that can bind to the virus and interferes with its ability to infect a cell. So in SARS-CoV-2, as shown in this image here, when there is a neutralizing antibody present, it will bind to the RBD region on the spike protein and block its interaction with the ACE2 receptor. So as a result, the virus is prevented from infecting the cell. So next, I would like to share the CDC's summary on the adaptive immunity. So as shown here, the immunity is a gradient. So the level of immunity can vary from prevention of severe disease, mild disease, and all the way to sterilizing immunity. So the main contributors to prevent the hospitalization, death, and also severe disease are the T cells, while the main contributors to prevent mild disease or asymptomatic disease are antibodies. And as shown in their conclusions slide, it is mentioned that neutralizing antibodies likely confer a majority, but not all immunity. So recognizing the important role of neutralizing antibody in preventing the virus infection, the FDA has issued a series of guidelines. So here I would like to show the guideline from development and licensure of vaccine to prevent COVID-19. So in this guideline, it is mentioned that for vaccine developers, it is required for them to measure the functional activity of immune response, which means that the neutralizing antibody that is induced by the vaccine will need to be accessed in order to determine its effectiveness. So here I would like to share the most recent fact sheet of the Moderna and Pfizer vaccine. So we can see that in the highlighted region, we can see that for both the Moderna vaccine and also the Pfizer vaccine, the effectiveness of the booster dose was based on assessment of neutralizing antibody titer. So how is neutralizing antibody measured traditionally? So here I would like to first introduce the conventional method, which is called virus neutralization test. So as shown in this image here, when there's no neutralizing antibody present, the virus will infect the host cell. And when there is neutralizing antibody, the antibodies will bind to the virus and prevent it from entering the cells. So by quantifying the number of infected cells, we'll be able to know the quantity of the neutralizing antibody present in the serum. However, this method is shown here. It requires the use of live virus and also cells. So as a summary, the advantage of this method is that it is gold standard due to its very high specificity. However, due to the use of the live virus and the cells, it requires very strict safety conditions, which is BSL-3. So this image here is the person. It shows the PPE you have to wear if you need to work in the BSL-3 environment. So it can be quite logistically demanding. And also, this test takes about 3 to 5 days to obtain the result. So it is quite tedious and slow. And due to the highly complicated experimental procedure, it also requires highly skilled operator. And also, this assay is very expensive and not scalable. And most importantly, due to the non-standardized protocol people use in different labs, the results are often not comparable between different labs. So to overcome these shortcomings, I would like to introduce this new technology called the surrogate virus neutralization test, abbreviated as the SVNT, that can also be used to measure the neutralizing antibody, but in a much more convenient and user-friendly manner. So as I mentioned earlier on, the neutralizing antibody binds to the RBD region and block its interaction with the ACE2 receptor. So based on this mechanism, the SVNT technology uses the purified RBD from the S protein and also the host cell receptor ACE2 to mimic the various host cell interaction in the ELISA plate well. So means that in SVNT, we use the protein-protein interaction to mimic the various host cell interaction. So in this assay, the RBD is conjugated with the HRP, and once the RBD binds to the ACE2 receptor that's coated on the plate, it will show a colored signal. And when there is neutralizing antibody present in the sample, it will form a complex with the HRP conjugated RBD, which will be removed during the washing step. So it will give rise to a much reduced signal. So by looking at the colored signal, we will be able to determine how much is the neutralizing antibody present. So this method is much more user-friendly, and it can be used in the BSL-2 environment, and the assays can be completed in 1 to 2 hours. So it is fast and also automatable and more suitable for high throughput applications such as mass screening. And also because of the standardized protocol we use, the between lab inconsistency is minimized. So although the assay is much more simpler and user-friendly, it still retains the high specificity because the assay permits the detection of all functional neutralizing antibodies that block the RBD ACE2 binding. And also this assay is independent of the isotype, and you can detect all neutralizing antibody, which is not limited to certain isotypes such as IgG or IgM. And also the RBD for this assay is present in the solution instead of coated on the plate. So this mimics the in vivo condition more closely. So how does the SVNT technology compare against the gold standard cell-based virus neutralization test? So here I would like to share the correlation data. So as shown here, so cPass is the commercial name for the SVNT. So we show that the results obtained from cPass and the PRNT, which is the cell-based virus neutralization test, is highly correlated. And we were able to obtain the R-squared value equals to 0.9446. And this R-squared value is very high, is considered very high in any biological system. So how about those assays, other commercial assays that is designed to detect the binding antibodies? So in this study, it is observed that for these 3 other commercial serology assays, their correlation with the gold standard VNT is very poor, with R-squared value that is less than 0.5. So this again highlighted that the SVNT is a more suitable technology for detection of neutralizing antibodies. So next, we also validated the sensitivity and specificity of the assay using the PRNT50 as a comparator. So as shown in this graph here, our positive and negative percent agreement are both 100%. And also because of the small volume required for the experiment, the blood sample can be collected using the finger prick method, and it also can be collected using the venipuncture method. So we have also established, tested the correlation between the two methods and showed a very good correlation. So as shown here, the percentage agreement here is 100% for either the finger prick method and also the venipuncture method. So because of the high sensitivity and specificity of detecting neutralizing antibody, the cPassTM assay is the first test that is authorized by FDA under the EUA for detection of neutralizing antibodies from recent or prior SARS-CoV-2 infection. And till today, the cPass assay is registered for in vitro diagnostic use in more than 10 countries, including the US, Europe, and also Singapore, Brazil, UAE, and Argentina. So we are still undergoing the registration in more countries. So the cPass assay is not only available as a research tool, but it is also available as a diagnostic tool. So for the general public who wish to test their neutralizing antibody, they are able to walk into a clinic and do the testing to see what is the neutralizing antibody level after they take the vaccination, and to get an estimation of the protection level. So next, I'm going to introduce how to interpret the cPass reading. So as shown here, the cPass reading can be expressed in percentage inhibition, which can be calculated using this formula here. So the higher the percentage inhibition indicates a higher level of neutralizing antibody. And we also have this cutoff value, which is 30%. So for the percentage inhibition above this value means there's positive of neutralizing antibody. So in addition to the percentage inhibition, the user can also, if the user needs to express the result in the antibody concentration, they can also do so by using the neutralizing antibody standard that is available from us to obtain the results on the concentration in U per ml. So this graph here shows the standard curve that can be obtained using the calibrators that is from the standard. And once you obtain the OD value for the sample, you can interpolate the concentrations using the standard curve. So what we have as an additional tool is the international standard for neutralizing antibody that can be used for calibration and harmonization of serology assays detecting the neutralizing antibody. So what shows here is a previous case, which is the hepatitis B. It is so for hepatitis B, the protective level of antibody is 10 IU per liter. And for the SARS-CoV-2, the WHO have also come up with an international standard that is the neutralizing antibodies to allow the harmonization of different assays. So of particular note, the IU per ml here is only reserved for neutralizing antibody. That means that only assays that is detecting the neutralizing antibody can use the IU per ml to report their results. And for binding antibody assays, they will need to report the results in binding antibody unit, which is BAU per ml. And because cPass is an assay to detect the neutralizing antibody, we will be able to report our results in IU per ml. So many of you have been wondering, is it possible to convert the percentage inhibition into IU per ml directly? So this will allow a more convenient comparison with other literatures. So to investigate this, a collaborative study was performed using 9 independent runs from China, Monaco, and Singapore to investigate the correlation between the percentage inhibition and the IU per ml. So in this study, using the WHO international standard as the calibrator, it is shown that the correlation between the percentage inhibition and the IU per ml is very high. And we observe a very consistent trend for the 9 independent runs conducted in different regions. And so this is the curve of all the data. And we are able to establish a mathematical model that allows the direct conversion of cPass percentage inhibition to IU per ml with a R-squared value of 0.977. So this method has been published in the Lancet Microbe, which is available for you to download. So next, I would like to share some of the quotes from the thought leaders in the field that recognize neutralizing antibody as a good predictor of protective immunity. So here I would like to share the quote from the Johns Hopkins University the National Strategy for Serology report. So it was stated that ultimately neutralization assays are the gold standard for determining if a patient has effective antibodies and protective immunity against SARS-CoV-2. And also the Dr. Anthony Fauci, who is the chief medical advisor to the US president, have always been advocating that neutralizing antibodies are the gold standard for protective immunity. So next, I would like to share some key takeaways from the WHO R&D blueprint consultation, where the experts meet to discuss establishment of correlate of protection that will benefit the process of vaccine development. So here it is mentioned by the experts that there is growing evidence show that neutralizing antibody responses could be useful in predicting vaccine-induced protection. And also the simplest option for any vaccine development is high-level, long-lasting neutralizing antibodies. So next, I'm going to share some of the key scientific publications that shows that neutralizing antibody has very good correlation with protective immunity. So in this publication that is published in Nature Medicine last year, they analyzed the mean neutralization level of the 7 commercially available vaccines with the protective efficacy that is observed in the real world. So as shown here, in this study, they analyzed 7 vaccines, so including the mRNA vaccines like Moderna and Pfizer, and also the AstraZeneca, which is the adenovirus vaccine, and also like inactivated vaccines like Sinovac. So as shown in this image here, we observe that with a higher mean neutralization level, we also observe a higher protective efficacy. So in the Moderna phase 3 efficacy trial, it is also reported that the level of neutralizing antibody is positively correlated with the vaccine efficacy. So some of the key message from this trial is that the vaccine efficacy is estimated to be 45 to 60% for vaccine recipients who actually don't develop any antibodies. And the vaccine efficacy can increase to more than 98% in recipients with the highest neutralization titers. And next, I would like to share a study that describes the SARS-CoV-2 outbreak in a facial vessel with a very high attack rate. So in this study, it is found that the presence of neutralizing antibody from prior infection was significantly associated with protection against reinfection. So as shown in this table here, they tested the 6 crew members using the Abbott IgG assay and also cPass, which is ACE2 binding blockade percentage. So as shown in this table here, we notice that all the 6 crew members are tested positive using the Abbott IgG assay. However, only those who are tested positive using the cPass assay are protected from the reinfection. So next, I would like to share some real-world data that indicates that the higher the neutralizing antibody, the lesser the chance of getting vaccine breakthrough infection. So the first study I would like to mention is an Israel study, which during that period, the alpha is the circulating variant of concern. So in this study, it was found that for those who are protected from infection, their neutralizing antibody level in IU per ml is in both IU per ml and cPass is much higher than those who have vaccine breakthrough infection. So in this study, it was shown that IU per ml of 250 and also which is equivalent to cPass 78% is a good suggests very high confidence against vaccine breakthrough infection against alpha. And also another study from Vietnam also investigated the neutralizing antibody among the healthcare workers who some of them are protected and some of them are have the vaccine breakthrough infection. So in this study, the main circulating variant of concern is Delta. So this study have two cohorts. The first cohort shows that with a neutralizing antibody level of 328 IU per ml, which is equivalent to cPass 82.3%, they are protected from the infection from Delta. And the second cohort showed a higher value, which is in cPass is about 90%. So this set of real-world data shows that a cPass value that is 80% to 90% actually suggests very high confidence against vaccine breakthrough infection. So we also noticed that when it comes to Delta, the required amount of neutralizing antibody to offer the protection become higher. So this is because for the variant of concerns, there are amino acid. There could be amino acid changes near the spike receptor binding domain, which could result in the reduction in the neutralization. So according to this study that is published in Nature, it investigated the neutralization of the wild type and also variant by Pfizer vaccinated samples. So as shown in this result here, so the gray bar is the neutralization level against the wild type, and the colored bars are the variants. So we see that when it comes to the variants, there's a decrease in the level of neutralization. So this is expected because we know that the vaccine is based on the wild type, original wild type strain. So when there's new variant of concerns, there is a decrease of the effectiveness of the neutralization level. So according to a summary from CDC, the neutralization loss can range from 0 to, for alpha, because alpha is very similar to the original wild type strain, to about 7-fold for beta. And luckily, now beta is not the main variant of concern right now. And for Delta, the reduction is approximately 1.5 to 2-fold. And I have also looked into some of the recent preprints that investigated the neutralization loss for Omicron. And the result reported are about 20 to 40-fold reduction. So this is also kind of expected because the Omicron variant has 15 mutations on the receptor binding domain. So many of you are interested to know that whether the SVNT technology can be used to detect the neutralizing antibody level against variant of concerns. And the answer is yes. So as I mentioned earlier, in the SVNT, we use the HRP conjugated RBD as the antigen. So this RBD in the kit is based on a sequence of the wild type, but we also provide reagents that is the variant, which can be used as a replacement of the wild type RBD in the kit, so that we can detect the neutralizing antibody against the variant RBD. So this is a list of all the different variants that we provide. So we have all the alpha, beta, and Delta, and also the most recent Omicron. So I would like to take this opportunity to also introduce the research tools that GenScript offer that can be used to accelerate your Omicron research. So firstly, I would like to introduce that we offer a different range of recombinant proteins for the Omicron variant, including the spike S1 protein, and also the RBD, and also the RBD that's labeled with HRP, which can be used together with the SVNT, and also the M protein. So these proteins can be useful for can be used as positive controls in assays, and also can be used as antigens for vaccine development. And also for the RBD, we do provide the binding data to ACE2 receptor. And we also provide different antibodies for the Omicron variant. So we provide a nucleocapsid antibody. So as shown here, we have tested its binding activity against the wild type Delta and also Omicron. And these antibodies can be used for development of LFA assays. And of course, we also offer the neutralizing antibody standard, which can be used to neutralize the Omicron variant. Next, we also provide plasmids that contains the S gene for the Omicron. So we provide the plasmids that is in the original codon and also optimized for expression in human cells. And for the vector, we also offer different selection of the vector, so you can choose based on your needs. And we also offer the peptide pool for the Omicron variant, which can be useful for T cell stimulation assays. And we also provide the pseudovirus for the Omicron variant, either as the pseudovirus itself, or it can also be provided as a pseudovirus neutralization assay kit. So these assays can also be used, is also widely used for detecting neutralizing antibodies, and it works in a very similar way that I mentioned earlier for the live virus cell-based virus neutralization test. But since it's using the pseudovirus, it can be performed in the BSL-2 setting, right? So last but not least, our GenScript also provides a different range of customized surveys to suit your need. So for the customized surveys, there's oligo and mutagenesis surveys, and we also provide customized recombinant protein and also customized cell line. So if you need any of these surveys, you can get in touch with us. So coming back to the SVNT technology. So just now I mentioned that we can replace the original wild type RBD with the variant RBD to detect the neutralizing antibody against the variant. So here I would like to share some data that we obtained using such method. So as shown here, for a patient who is infected with the wild type, it was found that the percentage inhibition of the wild type and alpha is the highest. And when it comes to beta, there's significant reduction. And for patients who are infected with the alpha, we also observed similar findings with a significant reduction of percentage inhibition against beta. And this is vice versa for sample that is infected with the beta. So this means that using the variant RBD in the SVNT, we are able to differentiate the neutralizing antibody response for samples that is infected with different variants. So next, I would like to share some data that we performed using the Delta variant. So here I would like to share this set of data that's obtained from Singapore. So in this study, we compared the neutralization level against the wild type and also the Delta. So for Delta, here the dPass is a nickname for the cPass assay, which is specific for Delta. So as shown here, we observe a high correlation between the cPass reading and dPass reading. That means that if a sample have a very high value against a very high cPass reading against the wild type, its reading against the delta is usually also high. And we also observed a reduction that range from 0 to 30% when it comes to the delta variant. That means that for the same sample, when it comes to the delta variant, we observe this reduction, which is also expected and consistent with other studies. So I would also like to share our study from Brazil. So in this study, we tested both the delta infected sample and the wild type infected sample. So we can see that for the delta infected sample, its percentage of neutralization is higher for the delta RBD, and it shows a significant difference from the wild type RBD. And while for the wild type infected sample, its neutralization against the wild type is higher. So this means that using this method, we are really able to differentiate the delta and wild type neutralization antibody response using this method, right? So next, I believe all of you must be wondering, how about for Omicron? So here I would like to share the recent study from Singapore that investigated that how effective is the mRNA vaccination against the Omicron variant. So as shown here, this graph shows the pre-boosting and post-boosting data. So for the pre-boosting, the recipients have received 2 doses of the mRNA vaccine. And for the post-boosting, they are boosted with another dose of the mRNA vaccine. So we see here that for the pre-boosting samples, the percentage of inhibition is very low. So most of them are negative, which is below the 30% cutoff. While for the post-boosting samples, there's a significant increase in the neutralization against Omicron, but we also see a very high degree of variability. And here is the comparison data for the neutralization antibody against the wild type, Delta, and Omicron for the post-boosting samples. So from this graph, we observe that for most of the samples, their percentage inhibition against the wild type and Delta is very high. However, some of them may still be not protected from Omicron. So this is also consistent with the observations that I mentioned in the first slide, where due to the onset of the Omicron, there's a drastic increase in the caseload globally. So here I would like to also share a study from US that is using the Omicron variant with the SVNT. So in this study, they also investigated the response of vaccinated samples against the wild type, Delta, and Omicron. So the vaccinated sample in this study is a mixture of the mRNA vaccine and also the Johnson & Johnson vaccine. So according to data sets here, we observed that for those who received 1 vaccine dose or 2 vaccine dose, and also 3 vaccine dose, or those who didn't receive any vaccine but got the natural infection, we can see that their neutralization for the Delta is lower than the wild type, while for the Omicron, it is the lowest. So this is also consistent with the Singapore study. And for those who have hybrid immunity, means that they received the vaccine and later on they also infected with the virus. So for such group of patients, we observe that some of them, they may still have a very high percentage neutralization against the Omicron, but this is not for all the cases. So this is also consistent with the Singapore study, right? So next, many people are interested to know whether the binding antibody assays are able to differentiate the different variants. So here, this is a comparative study that compares the SVNT assay and the binding antibody assay on their ability to differentiate between the Wuhan original wild type and also the alpha and beta. So as shown in this the graph here, we were able to show the differentiation for the different variants using the SVNT assay. However, for the binding antibody assay, it doesn't show any difference. So this means that the binding antibody assay can actually give a false signal that indicates a high level of antibody, but in reality, the functional immunity, which is the neutralizing antibodies, may have already significantly decreased against the variant of concerns. So here is a very good mechanistic explanation of why SVNT can differentiate, but the binding antibody assays cannot differentiate the variant of concerns. So in this study, we generated 4 monoclonal antibodies that have all of them have very strong binding activity against the RBD. However, only 2 of them have is able to neutralize the interaction between the RBD and ACE2 receptor using the SVNT. And these 2 are not even the strongest binders. So this again highlighted that if you need to detect neutralizing antibody, you will need to use the SVNT instead of the binding antibody assays. So next, I would like to introduce how can the SVNT be used for the 3rd generation vaccine development. So the 1st generation of the vaccine, which is all the commercially available vaccine right now, protect us against the wild type. And it is of urgent need to develop the 2nd generation of vaccine that can protect us against existing variants. But more importantly, it is also needed to develop the 3rd generation, which can not only protect us against existing variants, but also emergent variants and also like other SARS-related coronaviruses. So in this study that's published in the New England Journal of Medicine by the cPass inventor, they actually investigated the neutralization antibody response not only against the SARS-CoV-2 virus and also the SARS-CoV-1 virus. So as we know, the SARS-CoV-1 virus is the one that is responsible for the SARS outbreak in 2003. So in this study, here is the SARS-CoV-2 wild type and also the alpha, delta, and beta. And this one is the SARS-CoV-1 virus. So besides all these existing ones that is already circulated among humans, this study also investigated the pre-emergent subecoviruses, which is now still in the animal and may potentially transmit into humans. So because these subecoviruses, they are now still in the animal, the live virus are actually not available. So it is not possible to perform the assay using the traditional live virus method. So in such scenario, the SVNT can be a very powerful tool to use to determine the neutralizing antibody response for such cases. So how this is the result that is reported from this study. So as we see here, for healthy people who are vaccinated with the COVID-19 vaccines, we observe a very high percent inhibition against all the SARS-CoV-2 clade. However, the results, the percentage neutralization against the SARS-CoV-1 is suboptimal. So for those who have hybrid immunity, which means that they are first infected and then they also receive the vaccination, they show a higher percentage inhibition against the SARS-CoV-1 virus, but it's still not as high as the SARS-CoV-2. And most importantly, there is a group of patients who have recovered from the SARS in 2003, and later on during this pandemic, they also received the COVID-19 vaccine. So for this group of patients, they are actually protected not only from the SARS-CoV-2 virus and also SARS-CoV-1 virus, and also including the 5 pre-emergent subecoviruses. So this means that a high level and broad spectrum of neutralizing antibody level is observed for the SARS survivor who are vaccinated with the COVID vaccine. So this observation actually offer a very promising insight for the third generation vaccine development. So it is possible that we can reverse the order that we first vaccinated with the COVID-19 vaccine, and then the next vaccine actually can be based on the sequence of the SARS-CoV-1 that can potentially give us a very broad spectrum protection. So next, I would like to discuss the dynamics of the neutralizing antibody response and also the antibody longevity, because I believe most of you are interested to know how long can the antibody last in our body after the vaccination, right? So in this study that's published in Lancet, they investigated the neutralizing antibody response for those who have been recovered from the COVID-19. And as shown in this graph here, there are 5 different distinct types of profiles. So the first profile is the negative ones, which means they do not produce any neutralizing antibody after the infection, unfortunately. And then the second group is the rapid waning group, which means that initially they have a very high level, but then it decreased very fast. And at the end of the 6 months, it is become negative. And we also have a luckier group, which is the slow waning group. And most lucky group is the persistent group, which means that the speed of decreasing is very slow. And most of them still have a very high neutralizing antibody level after 6 months of the vaccination. So this means that the neutralizing antibody dynamics in recovered patients vary greatly, and the prediction of immune longevity can only be accurately determined at the individual level because of these huge differences we observed. So that one is for recovered patients. And how about those who have received vaccination? Do they also have a very highly variable neutralizing antibody response? So here I would like to share 2 studies. The first one is investigating the Pfizer vaccinated individuals. So here, shown in this graph here, we see that after the antibody peak 1 month after the first shot, we start to decrease over the next 2 months. And most importantly, we see the huge variation of the different data points. So meaning that it can really vary from very high to very low at all the 3 time points investigated, right? So the next study is to uses the SVNT technology to investigate the neutralizing antibody amount in Moderna and Pfizer vaccinated individuals. Here, we also observed a trend of decreasing over the 6 months post-vaccination. And most importantly, when we look at the arrow bar here, we also observe that there's a huge variation for different individuals. So how about inactivated vaccine, right? For inactivated vaccine, here I would like to share one study from Hong Kong and another study from Singapore. So the Hong Kong study showed that after the first dose of the Sinovac vaccine, most people are still negative, meaning that most people do not have produced the neutralizing antibody. And after the second dose, most people, there's a significant increase in the amount of neutralizing antibody, which is at a similar level after the first dose of Pfizer. And so the study from Singapore also reported very similar findings. So it is to our surprise that although these 2 studies are performed in 2 different regions using different sample cohorts and of course in different labs, the cPass value reported are very close to each other. So in the Singapore study, Sinovac dose 1, the cPass value is 16%, while the Hong Kong value is 15.6%. While Sinovac, after 2 doses of Sinovac, the Singapore study reported 66%, while the Hong Kong study reported 66.7%. So these values are very close, and the values are also very close for the Pfizer data sets. So this means that we can have very highly concordant cPass results from 2 different regions. So what I want to highlight besides this is also the highly variability for the different samples, especially for the inactivated vaccine group. So we can see here, after the second dose, the cPass reading can vary between 90% all the way to below 30%. So again, this highlighted the individual difference. So because of such individual difference, it is actually we have been advocating to have a neutralizing antibody guided booster dose booster shot regimen. So this means that on a populational level, we can do a mass screening using the cPass test and determine who are the ones that actually do not respond well to the vaccination. So so for those who have the cPass value that is above 68%, is considered high titer, which is acceptable for manufacture of high titer covalescent plasma. And those who have a cPass value that is above 80% is considered very high. So when we do a populational wide screening, we will be able to identify those who have low neutralizing antibody, and we can do the boosting for them so that we have lower chances of vaccine breakthrough infection. So this is more important for those countries who have a limited supply of vaccine. So when there's not enough vaccine for everybody, it is actually more helpful to vaccinate those who are not well protected from vaccine breakthrough infection. So next, I would also like to share the cPass performance, which is conducted by the WHO in their establishment of the WHO international standard. So in this study, they compared the between lab consistency for the gold standard PRNT and also the cPass SVNT test. As we can see here, for the PRNT, we observe a very different results across different labs. So the blue and red indicates the result of the same sample that is obtained from two different labs. So we can see the PRNT, we really see a huge difference for the same sample when the assay is performed in a different lab. However, for cPass, because of the highly standardized protocol we use, we are actually able to observe a very good consistency between different labs, as shown here. So this means that cPass can be the ideal toolkit for highly standardized neutralizing antibody testing. So here, I would like to conclude my presentation with the following points. So first, we know that only a fraction of antibodies produced post-infection or vaccination are neutralizing antibodies, and also the presence of spike protein or RBD binding antibody or IgG or IgM antibody does not mean that functional neutralizing antibody are present. And also, I have shared many scientific publications that shows that neutralizing antibody is a good biomarker for predicting protective immunity. This is also verified by many real-world data. So we also see that the neutralizing antibody wanes over time, and the waning speed and also the level of neutralizing antibody is highly variable between different individuals. It can depend on the type of vaccine you take. For instance, the mRNA vaccines usually induce a much higher neutralizing antibody level than inactivated vaccines. So it also depends on the age of the recipient. And apart from that, it also depends on the individual differences. So next, SVNT, as I showed earlier, can be the ideal tool for neutralizing antibody testing against pre-emergent subclinical viruses, which the live virus is not available to perform the traditional type of assays. And also we have observed a highly concordant cPass SVNT results across different labs and also across different regions. So I would like to end this presentation with the potential applications of cPass SVNT. So cPass SVNT can be used to determine the vaccine efficacy by measuring the amount of neutralizing antibody induced. And it also can be used to identify who and when may need a booster shot. And it is also a very powerful tool for next generation vaccine development. So this is the end of the presentation. I would like to thank for your attention, and I will be happy to take any question you have, or any exchange of comments or ideas are welcome too.